Greetings and welcome to the United States Transhumanist Party Virtual Enlightenment Salon. My name is Gennady Stoliarov II, and I am the chairman of the U.S. Transhumanist Party. Here we hold conversations with some of the world's leading thinkers in longevity, science, technology, philosophy, and politics. Like the philosophers of the Age of Enlightenment, we aim to connect every field of human endeavor and arrive at new insights to achieve longer lives, greater rationality, and the progress of our civilization. Greetings, ladies and gentlemen, and welcome to our U.S. Transhumanist Party Virtual Enlightenment Salon of Sunday, November 10th, 2024. Today, we have quite an eventful update for you about two recent events in the field of longevity science. First, I would like to introduce our distinguished panel for today. We have our Director of Visual Art, Art Ramon Garcia. We have our Director of Longevity Outreach, Ben Ballweg. We have our member from Texas who coordinates the activities of the Texas Transhumanist Party, Alan Crowley. And we have Dr. Edward Hudgens, who is the founder of the Human Achievement Alliance. So welcome to our panelists, and our special guest today is our very own Director of Scholarship, Dr. Dan Elton. Dan travels frequently to conferences and other events related to longevity, research into human aging, and approaches toward potentially defeating it. So, Dan, welcome, and please let us know what events you visited recently over the past several weeks.
Thanks, Gennady. Yes, I made a trip up to Boston. Recently, I moved down to DC for a new job at NIH, which I'll be discussing. But I made a trip back up to Boston for the Biomarkers of Aging conference. And the evening before that, Thursday evening, October 31st, there was a special, quote, Mortality Salon, which was hosted by the Foresight Institute and Longevity Biotech Fellowship. And it was held at a new office in Cambridge, Massachusetts, for an organization called Ethos. And Ethos is a AI startup incubator and AI center of excellence that's focused on AI ethics and sustainable development goals and a lot of other stuff. So the Ethos office space has been where a lot of really cool events have been happening in the Boston area. So I just wanted to mention them. They're doing a lot of cool stuff in the Boston area. They call it the center of gravity for AI in Boston. And that's really what it's become. So anyway, I'm going to go through some pictures I took. So when the evening kicked off, there was a series of talks, 4 talks total, of which one of them was mine. The first talk was by this guy Traden from the Augmentation Lab. Some of you might remember the Augmentation Lab from the previous Enlightenment Salon I did, where I discussed the Augmentation Summit. And he wanted to give a little bit of a alternative view on, and he claimed that he has never been afraid of death, which I think is a little bit unusual, because I think most people get some existential anxiety at some point. But he claims he's never experienced that. And he had kind of a theory about it, where he thought maybe it was because when he was young, his aunt was severely mentally ill or something, and kept saying she wanted to kill herself. So it's a bit of a crazy theory. But anyway, he gave a presentation which trotted out a lot of the arguments that we're familiar with about why death may be okay, because that people, people might get entrenched in certain ways of thinking. It's important to sort of call off the older people so the younger people can kind of rise up through the ranks and bring social progress and stuff. But he had another argument which was a little bit different than other arguments I've heard, where he was saying that human memory is pretty limited. So if you were, the longer you live, the more memories you accumulate, but you can't. There's some limit to how many you can accumulate, and so eventually you start forgetting things that happened a long time ago. So he was sort of arguing that even if you did live a really long time, the person you become isn't necessarily the person you were. So practically speaking, your identity is, because your identity is changing over time, it's not really lasting that long. Of course, there's counter arguments to all of these things that I'm sure most of you are familiar with. But so he gave the first talk. Then there was a talk from Nathan Cheng, who's one of the founders of Longevity Biotech Fellowship, and also, I believe, a co-founder of the Vitalist movement, and also a venture capitalist in Longevity Biotech. And he talked about some of the road mapping and initiatives they've done. He pointed out, up till now, there hasn't really been any plan on how we're going to tackle aging. Like, if you go to the National Institute of Aging's website and you look for a plan on how to fix aging, there's no plan at all. They just didn't even bother. So it's kind of ridiculous, really. I think he said that about 70% of people are dying from aging and aging-related illness, but there's not even a plan on how to tackle that problem. People just, So, so they decided to develop their own plan, and they've actually developed, I think, 3 different plans that are 3 different pathways towards unlimited lifespans and reversing aging. I don't have time to go into the whole thing, but one of them is basically fixing the damage. One of them is replacement. And then I think the third one was actually brain emulation, if I recall correctly. So he gave his talk was very interesting. And there was a lot of discussion of the replacement strategy, which some of you have heard about before, for instance, when we talked with Jean Hebert. And a really cool thing I learned, actually, is that Jean Hebert is going to become a program manager at ARPA-H, the DARPA for health. And so that's really exciting. And we might see some interesting projects around organ replacement, organ cloning. Nathan talked in particular about something I hadn't really heard discussed publicly very much before, which is whole body replacement. This is something that I think exists in some sci-fi. I forget the name of the show, but this exists in some sci-fi. The idea is that you would have a clone that would be essentially grown and kind of sitting in standby. It's a younger cloned body. And then you would basically do a head transplant where you would or graft your head onto this new body. It sounds kind of crazy, but I think this needs to be this approach needs to be one of the approach that's within the realm of discussion. And then so the only remaining major organ that would need to be replaced would be your brain. And Jean Hubert has actually talked about how you can do progressive replacement of neural tissue without losing memory or function. So you can do progressive replacements in theory. Now, if you're going to grow your own clone, to do this ethically, the clone can't be conscious, right? Because it would just be like growing in a tank or something, right? So you would have to genetically modify this clone so it doesn't develop a brain or anything beyond the brainstem. This sounds a little bit crazy, but he actually talked about how there's a condition, I believe it's called hydrocephaly, where essentially, what's it called? And there's a very strange genetic condition where, like, most of the brain is actually missing. In some cases, the people retain some function, but generally they don't have any function. So it actually seems possible that you could grow a clone that would just have a brainstem that controls vital physiology, but not any higher functions like consciousness. Anyway, so so then moving on to the next talk, that was by Alison Deutman from the Foresight Institute, and she talked about their tech trees, and they're continuing to develop the tech trees for different areas like longevity, neurotech, nanotech, and many of these are publicly available on their website. Then finally, I gave my talk, which I'll go over later. This was my Halloween costume. I was Bill Nye the Science Guy, and when I polled the audience, like most people didn't really know who I was, which is interesting because I also wore this costume around 2014 or 2015, and at that time, like almost everyone knew who I was. So it seems like the audience at this event was younger, and they were of a younger generation that hadn't really grown up with Bill Nye, which is kind of sad. But anyway, this was just a picture of me with Nathan Cheng. So now I'll show some pictures I took at the Biomarkers of Aging conference, which was at Harvard Medical School. And this was the breakfast the first day. I thought this was interesting because the breakfast was really unhealthy. And if they have a feedback form, I'm actually going to complain about this because I've recently adopted a gluten-free diet. So there wasn't really any good breakfast options for me. And furthermore, I recently learned that these paper coffee cups actually contain estrogen mimicking compounds. So when you pour hot coffee into these cups, there's actually a lot of like BPA like substances that leach into the coffee and can mess with your hormones. So turns out these paper coffee cups are not that healthy or good for you. Anyway, this is a look into the auditorium, which was completely full. The number of people at this conference has grown considerably since last year. So last year, it was the first year they ran this conference, and it was at the Buck Institute in the Bay Area. And I believe they had about 200 people last year. And this year they had almost 400 people, I believe, or around 350 to 400. I'm going to show just some random slides I took pictures of. This was one of the first talks, and there was a lot of discussion of both epigenetic aging clocks and protein-based aging clocks, or proteomic clocks. One of the big takeaways for me was that when it comes to predicting things like heart failure, heart disease, stroke, or just overall mortality, these aging clocks are actually not as predictive as I thought they would be. And so when you look at these, hazard ratios, they're generally not that high. So like what they do generally is they they will get a measurement of quote biological age from an aging clock, like an epigenetic clock, and then they'll compare that with your actual age, chronologically speaking, and then they'll look at that difference. So in some people, their biological age is greater than their chronological age. So it could be like 5 years or 10 years greater. Or in other people, their biological age is less than their chronological age. So they look at that difference, and then they can use that difference for risk prediction. And what they often do, like here in like this figure, what they do is they split the things into quartiles. So if you're in like the top 25% in terms of your chronological versus biological age difference, or biological minus chronological, either way. If you're in like the top 25%, then you have a certain increased risk. So like in this case, they're saying that you have a 2.86 times risk for heart failure within like 10 to 15 years. So when they give that sort of number, like saying you have a X increased risk within some time window versus the compared to someone like a normal control, that's called the hazard ratio. So hopefully that makes sense. But the big high-level takeaway I had from this conference is that these clocks are generally not that good for risk prediction. So actually, if you derive some measurements from CT scans or MRI scans, where you measure the plaque in your heart or the plaque in your aorta or how much visceral fat you have, I think those measurements could actually have greater risk prediction ability than some of these clocks. Now, where the clocks could be useful is for tracking changes over time. So a lot of longevity doctors like to use these clocks to monitor the effects of interventions. So, for instance, they might start somebody on rapamycin or metformin, and if you get these, if you get your biological or epigenetic age measured regularly, you can actually see patterns over time. So that's kind of what they're used for right now. But here's another example. This is actually proteomic aging clocks for I guess different proteins that are associated with different organs. This is just based on a blood test. Turns out there's tens of thousands of different proteins in the blood that come from different organs. So yeah, I believe that's what this was. This is looking at what things affect the epigenetic aging clocks, and one of the things that had the greatest effect in terms of reducing epigenetic age was metformin. So that's pretty cool. Metformin is extremely cheap medicine that's used for diabetes and I believe lipid control and stuff like that. This was, by the way, the speaker here is Steve Horvath, who is the person who developed the very first epigenetic clock. So he's kind of was like a superstar at this event. Now, it turns out the original clock that he developed doesn't really work that well. Like it can barely predict mortality, but it, the original Horvath clock does correlate with your chronological age to some degree. But since that original clock was developed, there's been dozens of other clocks that are much better. So then the field is rapidly evolving and new clocks are constantly being developed. I guess Steve Horvath had a recent paper, which is shown here, where they looked at methylation, which is a measure of the epigenome patterns. So they looked at the epigenome of different species, and they looked at the rate at which the epigenome was changing over time. So as you get older, the epigenome basically becomes more and more dysregulated. And consistent with the whole epigenetic theory of aging, they found that in animals with shorter lifespans, they tended to have faster rates of change in their epigenome. So that was the high-level takeaway from this work. Of course, the devil's probably in the details, but as with many of these things. But that's sort of the high-level takeaway. I believe this picture I took later on day one, And this was a talk by David Sinclair, who actually works at Harvard Medical School. So David Sinclair has been very interested in the epigenome and how the one of the things he's interested in is how the epigenome can be reverted to a more youthful state, which corresponds to if you rewind it back far enough, it can actually go back to like a stem cell. And so by reverting the epigenome back to the stem cell state, different organisms can regrow their limbs. So a famous example is the planarian worm, planaria, flatworm. Another famous example is the axolotl limb. Apparently, a mouse can regrow parts of its ear. And even in the adult human, we can regrow parts of our liver. Liver is a vital organ like the heart. So it's pretty cool. We can actually regrow parts of our liver if parts are damaged or removed. I think that's the only internal major organ like that. Now, I wanted to show a video now which summarizes David Sinclair's epigenetic theory of aging, which I thought was...
For about the past 50 years, popular theory has held that the process of aging is caused in large part by an accumulation of mutations. There's growing evidence, however, that aging has a significant epigenetic component. That is, the process by which stretches of DNA or the genes are turned on and off. My colleagues and I believe that not only are epigenetic changes a primary cause of aging, but that these changes are driven by the ongoing process of DNA breakage and repair. Why do we think this? Here we have two mice from the same litter, essentially twins. We checked, so we know that they have the same DNA sequence. The only difference between the two is that in the older looking one, the break and repair process was accelerated, and this caused epigenetic changes. Part of the epigenetic process involves the packaging of DNA. When genes are not active, they're packed tightly in chromatin, but sometimes this packaging loosens, giving the cell access to appropriate genes. This process happens all the time, and it has to for a cell to function properly. Certain areas of the DNA structure within a liver cell, for example, need to loosen to allow access to liver-specific genes. Unfortunately, naturally occurring DNA breaks cause changes in the chromatin structure, and over time, this happens a lot. Our bodies experience 1 million DNA breaks a minute, and as cells try to repair the broken DNA, the chromatin structure loosens in some areas, including the area around the break, and it tightens in other areas. And if it loosens in an area that it's not supposed to, a wrong gene might be expressed. This is important because if, for example, liver cells accidentally activate wrong genes, the organ as a whole will begin to show signs of aging. We wanted to test our theory that aging results from DNA break and repair, so we engineered a new strain of transgenic mice. These mice are genetically altered to let us turn on and off a special enzyme. When we turn the enzyme on, the DNA within many of the mouse cells are cut at a number of locations at a rate that is about 3 times the natural frequency of DNA breakage. And each breaking of the DNA initiates a repair. Special proteins that regulate chromatin structure and gene expression move to the cut DNA. The proteins loosen the chromatin and recruit other proteins that repair the DNA. When their work is done, the proteins usually move back to their original locations, and the chromatin structure, the packaging, usually resets to its original tightness. In our mouse, as long as the enzyme is on, cuts are initiated in the DNA. With many breaks happening, the repair proteins are very busy, and this is an issue. With each successive fix, the proteins don't always return to their original locations, and the chromatin structures at the repair locations don't always reset to their original tightness. Actually, the process is a bit more complicated. There are other proteins involved, and these proteins, in addition to assisting with DNA repair, are also responsible for turning genes on and off. With each successive fix, the proteins become more jumbled. This jumbling results in some genes that were initially turned on to turn off, and other genes that were turned off to turn on. Changes like these alter the cells enough to cause them to become more dysfunctional. By the way, just to be sure we weren't disrupting the function of cells, we didn't cut in random places. Instead, we avoided regions that code for genes. So what did we find? Here are the two mice I showed you earlier. They are both 10 months old. The older looking mouse had the cutting enzyme turned on for 3 weeks when it was young. The other didn't. To verify that aging we see isn't the result of mutations, we sequence, or read, the entire chromosome of each mouse. So really, the only difference between these mice is epigenetic. To test our theory further, we experimented with resetting epigenetic structures in our aged mice, and we found that we could safely reverse blindness and rejuvenate kidneys and muscle. We used to think that aging was like lines of a recipe being erased. Once a mutation occurred, the DNA code was lost forever. But now we know that cells still have the DNA intact. We just need to teach them how to read that code again. And if we can do that, aging may be much more reversible than we ever thought.
Okay. Great. Yes, thanks for posting the links in the chat, Janani. So I thought that was really good summary of the epigenetic theory of aging. And actually it's actually, you know, according to that video, it's sort of driven by the double-stranded breaks and having to repair those. And so he talked about that a little bit. This is Professor George Church, who many of you probably know, and he had a really interesting mask on during the conference, which you can see here. And this actually uses a HEPA filter. And there's actually a little fan in here which blows air. So you have positive pressure inside the mask. And this is actually really cool because it makes it a lot easier to breathe through, and you don't get any of the issues of CO2 buildup. So I actually bought one of these myself, which I have here. But as you can see, you can buy this on Amazon for about $50. It's called the Air Pro mask. It's pretty cool. So this could be useful for avoiding COVID and other airborne illnesses on like long flights and stuff. This is a picture lunch with Aubrey de Grey. Got kind of lucky and was able to have lunch with him. Got some updates on various initiatives like the LEV Foundation and the Astera mouse rejuvenome project and different things. This was just just another picture of the audience during George Church's talk. This was a talk that was all about using face photographs for biological age prediction. So just like with the epigenetic clocks, you can get biological age. You can also get measure biological age from just a photo of your face. And this might be useful for risk prediction. In this case, it was from cancer patients, but that was more of a kind of happenstance thing, because basically the only place that they could find a data set of these sort of pictures was from their cancer treatment center, because the cancer patients, because they're staying in the hospital, they get an ID badge, and for the ID badge they have their picture taken. So that was the data set they were able to get access to, which was I'm sure there's probably larger data sets available of pictures of people with their age, but this is what the one they had access to. This was a talk about ARPA-H. Actually, I'm going to skip over this because I'm not sure they want people sharing this, but basically they just were showing some basic stuff about prediction using different biomarkers and types of data. It's nothing too exciting, really. I'm going to skip forward here. There's this was a talk by someone from the Buck Institute, and more talks. This is kind of showing some different themes they observed when looking at how different things affect the epigenetic aging clocks. And this was a talk by someone from a biotech company, which is BioAge Therapeutics. And BioAge Therapeutics is developing a compound which mimics the effects of exercise. So it's called an exerkine, or exercise mimetic. And they had a lot of very impressive looking results. I sent it to my friend Greg, and he looked over these slides, and he was a little bit skeptical, but they have some exciting initial results on using this drug to treat obesity, but they also think it could potentially extend lifespan, and so that's kind of exciting. This is a picture of me with someone you may not be familiar with, but he's sort of a rising star in longevity. His name's David Barzilai. He's a longevity doctor in the Boston area, and he's pretty big on Twitter. And he has no relation to Nir Barzilai, who's the longevity researcher at NYU. But yeah, pretty cool guy. So this was a discussion where we had several famous longevity researchers gathered around for a demo of a platform being built by Aaron King, who used to work at the Foresight Institute. Aaron King was in charge of the Tech Trees initiative at the Foresight Institute, but he left Foresight Institute to found a company called Eon Biomarkers, which I'll be discussing a little bit later. And he had a demo of their biomarker visualization tool, and we were he was able to demo it in front of several pretty big name figures. Like here we have Nir Barzilai, David Barzilai, Ronald Primus and myself. So that was pretty cool. He got some good feedback on the visualization tool, because he's trying to figure out if the visualization tool would be more useful for clinical doctors or researchers. So here we had two doctors. We had David Barzilai and Ronald Primus, and we had Nir Barzilai, who's a researcher. Here's another picture of me with the two longevity doctors. This is the poster session. They had a huge poster session. And this is something I thought was pretty cool. So Aaron King, he had a poster, and this device measures your skin age using IR imaging. And so he was actually collecting data at this conference, which he's going to visualize on his platform. And so he was taking measurements on different people. And basically, you basically just hold your arm on this device, and it scans your skin and gives you a measurement of skin age. So this was me doing the measurement, getting a measurement taken. This is what the output on the display looks like. So this was my skin age. As you can see, it's right around the average for my age, which is 36. So I guess that's not bad. But what Aaron was doing was he was collecting all these measurements. So these are all the measurements he collected from the skin age reader during the conference. And the measurements of those little gray points, and then the average for different ages is shown in red. So anyway, you can see there's kind of a lot of noise in this data, but I mean, it's not like, it doesn't correlate that greatly with age, but there's a lot of variance. That's kind of interesting. Let me skip ahead. This is a very interesting slide here. This was from Nir Barzilai, and they have a new paper out where they identified FDA-approved drugs that are gerotherapeutics that they think could have some effect on longevity. One of the top was metformin, not surprisingly. And then there were several others. I never heard of SGLT2 inhibitors or biphosphonates. But some of the other people had heard of these. He also said that rapamycin probably would be higher on this list if there were better studies available. But there isn't right now. So I thought this was pretty interesting. So now I'll just give, do we have any questions? Do you want to take questions about the conference, Johnny, before I move on?
Sure. Yes, there are some questions about the conference. First of all, we have an interesting couple of posts from Calvin McCarter, who wonders how much of the recent emphasis on epigenetics in aging research is driven by the looking under the lamppost effect, in that we can undo methylation and chromatin aging more easily than mutations, and how much of this has flipped due to gene editing being more precise than epigenetic editing. So what are your thoughts on this set of questions?
Well, hey, Calvin. I think that the idea that the accumulation of mutations causes aging has largely been debunked. I mean, there's 2 copies of most genes. So my understanding is even if one copy gets knocked out, sometimes the backup copy can be, or the other copy can be used. And these cells have a lot of mechanisms for repairing DNA damage. I'm not sure about single point mutations, but I think what Aubrey de Grey says in his book is that just mutations by themselves don't have a huge effect on aging. As far as undoing the DNA methylation aging, what happens is that my understanding is that there's these compounds called sirtuins, which are help maintain the epigenome. But like was shown in that video, when these I believe these sirtuins are also recruited when there's double-stranded breaks to restore the epigenetic state after the double-stranded break. But what happens is the sirtuins, they get dysfunctional over time. So they kind of live on the, my understanding is they live kind of like on the DNA itself, and they have to kind of, when there's a double-stranded break, they have to go and repair it, and then they have to go back to kind of where they live, quote unquote. But they don't always go back to the right place. So they kind of become dysfunctional over time. So that's why there's interest in these kind of like synthetic sirtuins or like that sort of thing. I'm not really sure how successful that's whole thing is right now. But, David Sinclair is looking for small molecules which sort of, quote unquote, help the sirtuins. I don't think that's been very successful so far, but they're working on that. So I wouldn't say that this is, it doesn't seem like a looking under the lamppost type of thing. Maybe in terms of biomarkers, it is, because there might be some better biomarkers out there that, for instance, in proteins or something, but we just don't have the technology, whereas we have the technology for epigenomics. So that's my read on it. But he also had another question: how much of this is flipped due to gene editing being more precise than epigenetic editing? Honestly, I'm not the right person to ask, but I think gene. I think epigenetic editing is. It depends what you mean by that. Like, because I think precise epigenetic editing is still kind of beyond our reach. But there is a whole field of cellular reprogramming, which is kind of like moving the restoring, kind of reverting the epigenome back to like a stem cell state or a more youthful state. Rudy Hoffman asks, "What the heck is Church wearing?" Well, that was the mask. And he always wears a very professorial jacket or blazer. So, any other questions?
Yes. So we have a comment from David Wood, who writes, another argument against the looking under the lamppost explanation for the interest in epigenetic reprogramming is that reprogramming seems to unexpectedly reverse several hallmarks of aging. So thank you for that observation, David. And Rudy also wonders whether DNA methylation is the current state of the art in terms of biomarkers of aging. And he points out he is in the life insurance business, and he knows of 0 life insurance companies who use any advanced tests to evaluate their customers or potential customers' health. And I would agree with that observation in terms of my experience. Rudy also wonders, is anyone aware of institutional users of these leading edge tests? So if not insurers, any other large institutions that are trying to somehow deploy these tests and gather information about people's rates of biological aging, perhaps?
Yeah, like I said, one of the big takeaways for me was that most of these clocks are not that good at actually predicting mortality versus like other things. So, for instance, I think you could predict mortality based just on a few basic risk factors, almost as good as an epigenetic clock. A lot of these epigenetic clocks. So, like, for instance, if you just have some risk factors like your chronological age, BMI, smoking status, cholesterol level, and maybe a couple other blood biomarkers, I think that could actually predict mortality almost as good as a lot of these clocks. That's sort of my read on it. So it makes sense the insurance companies aren't investing in this right now. I see Ed Hudgens has a similar question. Yeah, he says, any insights on biomarkers that might be useful for FDA to use to judge the efficacy of treatments to slow or stop aging? Well, that's sort of what this whole conference was about. In fact, I think the whole impetus for this conference was the community realized we want to find and some consensus on biomarkers for clinical trials. And my understanding is the FDA is actually open to the idea that you could have a biomarker for aging, and that would serve, and reducing that biomarker or changing the biomarker would serve as an endpoint for a clinical trial. The FDA is open to this. There's just right now a lack of consensus on how to do it, and so there were a number of different approaches presented. A lot of them were epigenetic clocks, but there are also proteomic clocks that look at thousands of different proteins in the blood. And there were a couple presentations and posters on imaging-based clocks. So, but not a lot of consensus, but the field is developing, and people are actually starting to talk about combining multiple different clocks. So it's all rapidly evolving, there was a poster showing that some of the newer clocks are more responsive to interventions. So, for instance, there's been studies on what happens if you adopt a Mediterranean diet, for instance, or what happens if you start taking metformin. And there was a poster showing that some of the newer clocks have a larger effect. There's a lot of variability. So like on the individual level, there's a lot of noise and variability in how these things change with over time. But when you start to collect data on, larger groups of people, you start to see statistically significant effects from interventions with the newer clocks. So I think that's pretty encouraging. Yeah. Any other questions?
Yes, and Edward has followed up with a comment that FDA is open to this idea. There are opportunities in the next few years to give this greater priority. So thank you, Edward. There is a question yes, go ahead.
Well, before that, I noticed Jason Geringer and was mentioning stress, and a couple other people mentioning stress. And that is something that comes up, which is that acute stress seems to increase biological age on these clocks. But the really cool thing is it's actually reversible. So for instance, pregnancy increases biological age, but then it seems to go away after a year or so. And there was other other acute stressors could have the same effect, but it's the heartening thing is that it seems to be reversible.
Yes, indeed. And it's interesting also that you mentioned some of the work being done at ARPA-H. I happen to know that Jean Hébert did start in his position as a program manager at ARPA-H. And again, I would encourage everyone to watch our Virtual Enlightenment Salon with him from April 11th, 2021. And we also had a panel with him that I hosted at RadFest. It is called the Future Makers Panel, and this is a panel where he presented in late 2022 an updated perspective on the research for brain replacement, but now he is going to have a sizable amount of funds, an 8-figure sum to allocate essentially to do some serious trials in the efficacy of the incremental brain replacement approach that he outlines. So this is quite exciting news. I would say in terms of evaluating various events that happened during the Biden administration, I think people know that I have not reserved any criticism when I've had such criticism, but I think the founding of ARPA-H was one of the bright spots during those 4 years. And ARPA-H could make a huge difference in terms of the rate of progress in anti-aging research, especially with people like Jean Hébert among the program managers. But Mike Lezine wonders, why would ARPA-H want to hide anything? So you mentioned a presentation that maybe they weren't as interested in popularizing, given especially that this is a federal agency and there has been more of an emphasis on open access to data generated by federally funded research recently. it doesn't seem like federally generated research output would be copyright protected per Mike's comment. So what do you think is behind the apparent secrecy or semi-secrecy?
Secrecy about what? What they're the slides? I don't really know. By the way, the first day of this conference, they said that we were not allowed to take pictures of the slides because they wanted to encourage researchers to be very candid and open and share their latest results and conjectures and stuff. For that reason, a lot of the presentations I wasn't able to take pictures of, but a lot of the presenters said that it was, and then the first day they said it was okay to take pictures. The ARPA-H talk was actually, I think, the second day. The second day, they said everyone was allowed to take pictures. But as you saw on the slide, it actually says, like, on the bottom, it says confidential material. So I don't think there's anything super confidential there. But I wouldn't read too much into it.
Right.
But here's the thing. Well, I actually talked to the guy who gave the talk. Let me see what his name was. He was so the guy who gave the talk at ARPA-H is going to be starting as a program manager soon. And let's see. Let me see if I can get his name. Just give me 1 second.
Right. And while you look that up, I will note sometimes confidentiality language gets put on slides or other documents as essentially boilerplate. So there is an inclination to overprotect material sometimes as an institutional default.
So his name was Andrew Brack. He's from Liverpool, England, and he's a professor in the Department of Orthopedic Surgery at the University of California, San Francisco. And he's it looks like his research specialty is stem cells, which obviously is related to aging. So he's I'm going to put a link in the YouTube chat here. Yep. Gennady found it. Yeah. So I got to talk to him briefly along with Aaron King, and we were actually showing him the biomarker visualization platform. Anyway, what he told me is that he's being very careful about what he says right now about ARPA-H, about this new program he's leading at ARPA-H, because he just joined a few months ago, and the call for proposals has not actually been put out yet. So he's being very careful about talking about it because he doesn't want to give anyone an unfair advantage when it comes to submitting proposals. Does that make sense? So that might have been why they wanted those slides to be a little bit confidential. I'm not sure. I don't think so. I don't think that's the reason, but that was a reason that he couldn't really go into any specifics with us about the program. Yeah. So let me. Oh, Rudy Hoffman's asking, what is ARPA-H? Well, this is the Advanced Research Projects Agency for Health. It's modeled after DARPA, at least loosely modeled after it. And it's actually located within NIH, but it's a separate institute that is actually separated from the NIH director and the NIH org tree. So it's kind of its own separate institute, but it's considered part of NIH. And the reason they did that was so that they could utilize a lot of the systems already at NIH, like the human resources systems and human resources department and things like that. So there was a lot of debate about whether it should be a completely separate institute or whether it should be within NIH as a sub-institute within NIH. So they kind of found a middle. They sort of found a compromise path there.
Yes, indeed.
Yeah. So why don't I go on and give the rest of my slides? So I have some additional slides, which are some of them are slides that I gave at the Halloween event in Cambridge. So let's get into this stuff. So a lot of the motivation for the research I've done is that today healthcare is actually sick care. So instead of proactive preventative care, we have reactive care. And only the very wealthy can afford true preventative health care that is tailored and personalized to them. And I think a big reason for this is just the doctor shortage and the way that the American Medical Association has capped the number of medical degrees that can be granted in the United States. And they've overly restricted the granting of medical degrees. So now we only have 2 to 3 doctors for every 1,000 people. And meanwhile, what's happening is the amount of data that's being collected on people is growing greatly every year. In some cases, it's growing exponentially. So for instance, in radiology, the number of medical scans is increasing about 6% per year, but the number of radiologists is not increasing every year. So it's predicted that in terms of the number of images that radiologists are going to have to look at, they're going to have 40% more work by 2030. And so what's happening is all this data is being generated and a lot of it's not being utilized much. This includes imaging data, EHR data, and genetics data. So this is where I think AI can bring the most value, not by replacing doctors completely, but by filling all these gaps in our healthcare system. Another thing that motivates me is this very convincing argument that through 80 to 90% of heart disease today could be prevented if we can identify very high-risk individuals early enough. And this argument is based on autopsy studies of young individuals. So this is something I talked about in a previous Enlightenment Salon, but I'm going to go over it again. Their autopsy studies show that about 15% of 15 to 34-year-olds have these fatty plaque streaks in their arteries. And these are the earliest stage of atherosclerosis. And it's believed that these are largely due to genetics. So if we can identify these high-risk individuals when they're very young, like in their teenage years, and then we implement a very aggressive interventions like statins, metformin, GLP-1 agonists, and gene therapy, then I think we can prevent 80 to 90% of heart disease, which is pretty exciting. So a lot of my previous research work from years ago was on developing these tools for doing automated measurements from CT scans. And we had this paradigm which we call opportunistic screening. So in opportunistic screening, when you have a medical scan taken like a CT scan or an MRI scan, you have AI models that run in the background, and they do all these automated measurements, and they look for certain conditions. So with all these measurements, you can do risk prediction for cardiovascular disease, diabetes, all sorts of other different conditions. And then this risk information can be provided to the clinician and patient. So with these different measurements, we can visualize how people compare with other people in their age and gender cohort, and then we can identify outliers and flag those individuals for follow-up and things like that. So this is just a kind of prototype dashboard of how a lot of these measurements could be displayed to clinicians. So just to go over it again, we have measurements of bone mineral density, plaque in the aorta, visceral subcutaneous fat, muscle, and liver volume, liver fat. The most important of these measurements in terms of predicting cardiovascular disease risk is the plaque in the aorta. This is showing a sagittal view of a CT scan, and the plaque in the aorta has been segmented here in green. So I helped develop a tool that automates this segmentation. And to get a standardized measurement, we kind of measure between these two vertebral levels. So the other thing I worked on years ago was predicting diabetes risk from CT scans by looking at the morphology and structure of the pancreas. And this is the first time that this had ever been done before. Since then, other researchers at NIH in the same lab have developed that work further. And now, to predict diabetes risk, they're not just looking at the pancreas, but they're also looking at the blood vessels and arteries that are feeding into the pancreas. And they're also looking at the fat that's around these arteries. And they're finding that the buildup of this visceral fat or perivascular fat is a risk factor for diabetes. And also, if the intensity of level of this fat in the CT scan can actually be a measure of inflammation within the arteries. And so the X-ray intensity or attenuation of the fat around the arteries can be used for diabetes risk prediction and also cardiovascular disease risk prediction because it's sensitive to the inflammation. Now, more recently, we ran these tools on 200,000 scans at Mass General Brigham, and we got a huge data set. And as I mentioned before, Aaron King has a startup called Eon Biomarkers, and he allowed me to use his tool for, and he shared the code base, so I could actually visualize the CT image biomarkers from 200,000 scans using his platform. You can find this you can find this at agingbiomarkers.info. Since we have time, I'm going to just do a short demo of it. Let me just share. So this is the biomarker exploration tool that Aaron King and his team developed. And there's more to their software as well. But this is just the what they've released publicly, which is the visualization component. And there's a public data set of blood biomarker that's been uploaded here. As you can see how these biomarkers change with age. And what Aaron has done is he's plotted both the mean and the median, so at different ages. So the mean is in black and the median is in red.
Yeah. This is about how to cure aging. Where did you want to go?
What was that, Ed?
Trader.
Oh, okay. So the reason they do this is because when you have the median kind of splitting away from the mean, that means that it means that there are outliers, or there's a subpopulation that's sort of splitting. And so I actually said that wrong earlier, but the mean is in red, the median is in black. So what you see sometimes is the mean actually splits away from the median, and that's because the mean is more sensitive to outliers. And also, if you have a subpopulation that's kind of splitting away, you'll see that more in the mean rather than the median. So one thing Aaron likes to do is he likes to look for when the mean is splitting from the median. The other thing he likes to do is he likes to look for these inflection points. So, for instance, with blood pressure, you see that it goes up during early development, and then it sort of plateaus for a while. And then there's sort of an inflection point around age 35, 36, 37. And right now, I don't think we have a very good understanding of why these inflection points exist, but they're definitely interesting to see. Now, the other thing that Aaron likes to talk about, which is unique to this platform, is that you can select subpopulations. So let me show you a demo here. So if I go, these are all the biomarkers that you can visualize from open source data. So let me show you a little demo here of what I'm talking about. So if we select glucose, plasma fasting glucose, what we can do is they have this highlight points tool, and we can actually select all of the people with low glucose if we want. And then what happens is that all of those people we selected, their data points are highlighted on all the other graphs. So sometimes you can see interesting patterns in these subpopulations using this type of tool. Uh, anyway, so feel free to check this out. As I said, I also visualized the imaging biomarkers here. And we're hoping that someday we can make all of those data available on this same website. So,
Excellent.
Well, I'll show you some graphs of the imaging biomarkers, which from when we ran on the 200,000 CT scans. So here we're looking at the non-contrast CT scans in particular, because when you have the intravenous contrast applied, it can interfere with the measurements. So in these plots, we'll be looking at data from around 39,000 CT scans. And so here's the data for the bone mineral density. And as you can see, it just sort of linearly declines with age. This is really nothing new. This has been shown before, but it's nice to see. Now, this is more of a new finding, which is the aortic plaque as a function of age. So we measure both the volume of the plaques and the density of the plaques. And there's a way of scoring the severity of the plaques, which is called the Agatston score, which is just kind of an arbitrary metric, but it's the kind of the clinical standard metric. So this is the Agatston score for plaques in the aorta. And you can see that I use a logarithmic scale on the y-axis. And you can, this sort of thing has been measured before, but I don't think anyone has ever plotted this sort of data over such a large range of age. So it's kind of interesting. And what you see is that a number of things. So first you see that a lot of people have zero plaque, even as they get older. There's a significant number of people who just have zero plaque, even as they get into their 80s and 90s. But then there's a very significant number of people who start to get plaque, and it seems like there's a sort of inflection point here around age 40, where the level of plaque starts to increase. And it's not exactly exponential, but it's close to exponential. So if it was exponential, it would be a straight line. It's obviously not a straight line, but it's somewhat straight through, like age 40 to 60. Yeah, so this is kind of interesting, mostly because of the wide range of ages we were able to plot this over. This is looking at the fat area. This is another sort of different type of trajectory here, where fat area kind of increases until about age 40, and then when you get very old, it actually starts to decrease. There's a similar story for visceral fat. And also you see that males have much more visceral fat, which is not surprising. Men are known for the beer belly and that sort of fat deposit. This is muscle attenuation versus age. So this is like muscle density versus age. And what's happening here is that there's fat that is infiltrating into the muscle tissue and decreasing the density of it. And that seems to kind of go just kind of linearly with age for whatever reason. So those are some of the graphs. There's some of the more interesting graphs from the new data set. So then, do we have any questions about that?
I think this is a fascinating aggregation of data. My question is, I understand that the data extends to age 90, but of course not all individuals in the data set survive to age 90. And you pointed out that it's possible to isolate certain data points for certain individuals. Have you tried looking at people who survived all the way to age 90 and what their patterns for various biomarkers over time were?
Yeah, so we actually had quite a few scans from people that were older than 90, but unfortunately when we anonymized the data, we had to basically remove their age because there was a concern that if you have someone who's very, very old, like 99 or 100 or something, then they potentially could be identified just based on the age and the fact that these were scans come from Mass General Brigham. So, yeah, so unfortunately, when I made these plots, I didn't have access to the raw data. I only had access to the de-identified data. So I didn't have the ages greater than 90. But, and unfortunately, because I've left that job, I don't think it would be able to get that data, but get those ages at this point. But to answer your other question, that's something I'm going to be exploring, actually, is I'm interested in looking at the medical images, CT and MRI scans of very old individuals to see if there's any interesting patterns. And so that's one area of possible research I'm going to be exploring as part of my job here at NIH. But as of right now, I don't have anything to say on that.
Right. And hopefully some of your former colleagues might be able to have access to the data for the very old individuals, and perhaps you might suggest to them that this is something they should look at, because what is interesting to consider is, do these individuals who survive to the more advanced stages have different patterns regarding what their various biomarkers were when they were younger? And to what extent does it help to, if you can affect those biomarkers to some extent, to keep them either in a more optimal range or even, say, toward the better end of the optimal range, whatever that might be?
Yeah, unfortunately, we don't have a lot of data from the same patient over time, or what they call longitudinal data. So there's about 39,000 scans graphed here. The results of the measurements from are graphed here. And those 39,000 scans come from like 34,000 patients. So most patients only had one scan. There were a couple hundred patients that had two or more scans. And so I tried graphing how things changed among those patients that had two or more scans. And what I saw was there was a lot of variability. And it was actually kind of interesting because like when I plotted the plaque over time for patients who had more than two scans, in some patients the plaque increased, the amount of plaque increased. In some patients it was around the same, and then in other patients it actually decreased. So yeah, you could, if you're able to get this sort of longitudinal data, you might be able to see interesting trends where maybe in the very long-lived individuals, for instance, they have actually doing something that allows for the plaques to regress over time, or something like that. I don't know, but it would be interesting to look at.
Absolutely. It would be fascinating to find that out. But yes, any more questions on the data from agingbiomarkers.info from anybody on the panel? Feel free to ask now. And if not, then we can proceed to the next portion of your presentation, Dan.
Yeah, I just have a few more slides just touching on some of the research plans that I'm developing in my new role, the National Human Genome Research Institute. And as a disclaimer, I'm just speaking here in my personal capacity. Nothing I'm saying should be construed to represent the NIH or the federal government or anything like that. So one thing I'm hoping to look at is what we can do if we combine genetics data with imaging data. And if we're able to combine whole genomes with like medical images, then we can start to find correlations between genes and biomarkers that are measured from the medical images. There's been a number of papers on this, but not that many. Like I think I found about 5 to 10 papers. And almost all of them are using the UK Biobank. We're hoping to create a data set here at NIH, which combines both of these. We're working with an initiative called the All of Us Initiative, which is sort of like a US analog to All of Us. To Biobank, UK Biobank. But as of right now, the All of Us program hasn't actually collected any imaging data. So we're hoping to push them to maybe start collecting more. I have heard they're interested in that, but they haven't actually collected any yet. Anyway, this is an example of some research that was done using the UK Biobank, and they were looking at these DEXA scans, which are dual energy X-ray scans. They're actually just they're not really scans. They're more like just images. It's basically just an X-ray image that's taken at two different energies. Anyway, they had these DEXA images, and what they showed is you can actually get a measurement of the plaque in the aorta from the DEXA scan. And so the plaque is kind of these bright splotches in this red region here. And they had a deep learning model which automated the measurement of the plaque. And so they were able to find genes that are associated with high amounts of plaque. One of them was this famous APOE gene, which is implicated in Alzheimer's disease. And then there was a couple other genes. So this is the kind of work I'm hoping to do, but I'm hoping to do this sort of work using CT scans, where we can get more precise measurement of plaque, and also we can look at the structure and volume of organs with CT scans. So it's become very easy now to segment organs in CT scans and get very accurate measurements of organ volume and organ density using in an automated way. So there's a open source model which is called the total segmentator, and it can actually segment out 118 different structures in CT scans. So we're hoping to use this, and one little complication is that sometimes not all of the organ is visible in the CT scan, because, for instance, with the chest CT, they might only take it in a certain region to minimize X-ray exposure. But we have ways of dealing with that. There's other AI tools that can actually determine whether the entire organ is present or not, and what organs are present. So, but using these different AI tools, it's become extremely easy to get very precise measurements of all these different organs. So we can start to look for genes that might affect like the size of organs and things like that. There's one study I found that looked at genes that are associated with the structure of the aorta and the diameter of the aorta. So that might be interesting. Now, but going back to the plaque, it occurred to me that if we can predict the risk of plaque from genetics, that actually might be more useful from a longevity perspective than predicting cardiovascular disease. Because it's sort of, I think that if you're interested in living very long and living to like 150, for instance, using longevity biotech, it's much more useful to know if you're high risk for plaque than cardiovascular. Like if you're going to be, it would be much more useful to know that in your 80s, for instance, you would be at high risk for plaque versus cardiovascular disease, because now, right now, that doesn't. Right now, the risk models. So right now, the risk models don't really tell you about plaque. They just tell you about your risk for a major event like cardiovascular, like heart failure, stroke, and aortic aneurysm, and other things that are lumped under the term cardiovascular disease. When we talk about cardiovascular disease, what that actually means is there's like 5 different types of major events that can happen. that are logged in hospital databases. And that's what we mean by cardiovascular disease. Plaque doesn't, you can have a lot of buildup of plaque in your heart, but not be considered to have cardiovascular disease. You need to have one of these major events, but I think from a longevity perspective, it's very useful to know if we can predict if you're going to have this plaque, because it may not be an issue when you're 80 or 90, but it could be an issue later on when you're like 110 or 120, right? Does that make sense?
Yes, it does.
Yeah. So I think this could be very useful. Final thing I'll talk about just briefly is risk prediction using genetics. So there are a lot of these conditions we're interested in, like cardiovascular disease, diabetes, Alzheimer's, dementia, etc., have a large genetic component. Often like 40% or 50% of the variation between individuals can be just explained or associated with genetics. However, there seem to be many, many different genes that are involved. So that's why to do risk prediction, you have to look at really all of the genes. So you have about 20,000 genes. And so when you do risk prediction using all of the genes, actually to be more specific, what you do is you look at different variants. So there could be, there's specific genetic variants called SNPs, which have been found to be associated with different types of outcomes and have been found to be important. And there's I think there's about 100,000 of them. So anyway, you want to do risk prediction using like 100,000 variants. And that's what's called polygenic risk prediction. And then basically they use a linear model, which might sound kind of silly because we know that the genome and the way genes are turned on and off and how the different genes affect each other is very nonlinear. But it turns out that these linear models actually work really well. And there's kind of evolutionary arguments why this is the case. There's these linear models that are being developed that predict different types of diseases. And you can also predict things like height, for instance. This is showing prediction of height and how the predicted height compares with the actual height of people. So this is something else I'm interested in. There's already been a lot of work in this area, but I'm hoping to possibly do something in this area as well. And then more generally, we're also looking at just improving the data platforms at NIH. And the final thing I'll mention that I'm going to be working on a little bit just as kind of a side project is kidney stone risk prediction from CT scans. Turns out if you look very closely at the kidneys in a CT scan, people who are at risk for kidney disease and kidney stones, they get these they start to get these deposits of minerals and stuff inside their kidneys. And there's been a small amount of work showing that you can actually detect these deposits, which are the precursors for kidney stones, in a CT scan. And the way it manifests itself is just a slight increase in the density within the kidneys. So we're hoping to build a automated AI tool that will detect these subtle changes and predict risk of kidney stones. Currently, radiologists do not look for this, and they do not alert people if they're at high risk or not. But AI could do that. So thank you. That's all the slides I have. And happy to take any questions in the remaining 15 minutes.
Yes, well, we have about 8 minutes remaining, but thank you for the fascinating presentation, both the overviews of the Immortality Salon and the Biomarkers of Aging Conference, as well as your own research in progress. Edward Hudgens wonders, at the Biomarkers Aging Conference, were there any FDA people?
That's a great question. I did not see any. I don't think. My guess is there probably were not, because probably I would have bumped into them or seen them if there were. But
I asked that because I've attended FDA conferences and brought up issues like this one-on-one with some of the people there, and it's kind of disappointing that the FDA hasn't plugged into what is going on, because they are open, in fact, to biomarkers for aging. And I think that in the future, and this is for a wider discussion that we won't have here, there's going to be a lot of interest in exactly this with the new administration. I can talk, offline or even in one of these sessions about what's going on down here in the DC area. So I think we really have an opportunity to do that. And I'm a little disappointed that the FDA is still so insular, though it doesn't surprise most of us on this this Zoom. And you're back in the DC area now, right? Where are you physically? I'm just curious.
I actually moved back to exactly where I was living before, which is North Bethesda.
No, yeah, I know where it is.
Very close to you.
I'm in Rockville. Okay, I got to get off in a few minutes, but you and I can get together offline and have coffee and plan and plot longevity and so forth. So anyway, and Gennady, thanks for this. This is a great forum and a great presentation.
Yes, thank you, Edward, for your thoughtful questions as well. By the way, Edward has shared his article, which is entitled From Sick Care to Health Care and Your 200th Birthday. So I would encourage everyone to read that on the Human Achievement Alliance website. And another recommendation from Edward is this book, ChatGPT MD, which was co-written by Dr. Robert Pearl and ChatGPT. So there are some applications of AI now, even large language models, to medicine. And along these lines, Art Ramon has wondered whether Dan is allowed to use AI tools at the NIH, and clearly Dan's presentation shows that he is using AI tools, but there's a more detailed question. What kinds of AI tools, and are you only using in-house AI tools, or are you allowed to use external tools? Like, is there some sort of way in which ChatGPT might be able to assist, or other large language models, in terms of advancing this research?
Well, currently, I'm just getting up to speed with my research, so I'm not. I've only been here a few weeks, so I'm not using anything right now. But I'll likely be using that total segmentator tool, as well as some of the other biomarker extraction tools, which were developed in-house. So that when I worked at NIH previously, I developed a lot of those segmentation tools that I showed. And we've shared those with University of Wisconsin and they actually kind of packaged them together, and then they shared them with Mass General Brigham and other places. So those have been shared around a bit, but they're not public. Yeah, we can run AI segmentation models here. As far as ChatGPT, I mean, I have access to it. As far as like API access, I don't have API access, but apparently there is an NIH initiative called Strides where researchers can get various things, like, which I think might include API access. As far as whether they're using AI here at the NIH Clinical Center, my understanding is they are not. But I think there are AI tools that are useful in healthcare. But generally, a lot of hospitals don't have the manpower expertise to deploy them right now. GPT-4o was recently showed to be quite accurate at answering questions about medical images. There was actually a study that was published recently where they collected a series of questions about medical images. There's a website where they have a question of the day for radiologists, where they show an image and they have some question. So they collected all these questions, and they found that GPT-4 was about as accurate as your average radiology resident. So that's like very impressive, because GPT-4o wasn't specifically trained on medical images. If something like GPT-4o was trained on all of the medical images within a large healthcare system like Mass General Brigham, and it was trained on all those images and the radiology reports, I think it would be easily as accurate as your average radiologist. Now, there are some specialist radiologists who are like really good at particular things, like, like there's some that are really good at looking at prostate MRI. There's some that are really good at looking at chest CTs and stuff. So maybe it wouldn't be as good as those really skilled radiologists, but I think it could easily be as good as your average radiologist.
Yes, thank you very much for that answer. And indeed, the progress in AI capabilities has been quite impressive over the past 2 years. I have definitely noticed significant increases in analytical reasoning abilities, the ability of the large language models to do math, higher level mathematics correctly, which is quite noteworthy. Yep. And hopefully these tools will become more available to NIH researchers as well. So
yes, well, I think what you're referring to is the new what's it called? So the new ChatGPT tool, which is
GPT-4o.
So there was something that was codenamed Strawberry, and it's called GPT-01.
GPT-01.
So
okay, so I think in the next in a subsequent salon, we're going to need to explore GPT-01 to a greater extent. Unfortunately, we're out of time right now, but this has been a wealth of information. Thank you very much, Dan, for your thorough presentation as always, and I hope we can benefit from all this research and live long and prosper.